Hi, I'm Kirthika Elangovan and I'm currently pursuing my Masters in Microbiology at CUTN, and I'll be talking about a few advanced bioremediation techniques in this video. So before we get into the techniques, I just wanted to brief you all in on what bioremediation is. So microbial bioremediation is an environmentally sustainable approach to manage pollutants. What makes microbial bioremediation superior in comparison to other remediation techniques is the microorganisms, adaptive versatility, and wide metabolic repertoire. And with the advancement in molecular biology and technology, we are able to get a detailed and holistic view of all of the microbial communities and their capability of carrying out bioremediation. First up, we have biosorption, which relies on the ability of biological materials to passively absorb and bind contaminants onto the cellular structures. Unlike bioaccumulation, which requires metabolic activity, biosorption is metabolically passive, so the process can occur with both living and non-living biomass. And biosorption involves or occurs through three steps. Initially, we have contact between the pollutant and the biosorbent. Next, we have absorption of the pollutant onto the biosorbent surface through physical forces that can either be van der Waals force, ion exchange, or chemical interactions. And the final step is equilibration, where the pollutants are bound and reach a stable concentration on the biosorbent. Coming to the examples, there are several bacteria and several fungi that carry out bioremediation using biosorption, but then I'll be talking about only a few. So first we have Pseudomonas, which is a versatile bacterium that is widely used to degrade heavy metals such as chromium, cadmium, lead, and arsenic. Then we have Phanerochaetate Chrysosporium that is capable of degrading polycyclic aromatic hydrocarbons because it is known for the secretion of lignin degrading enzymes that will help break down these complex contaminants into simpler molecules. And then we have the fungi Penicillium chrysogenum that is capable of degrading lead and cadmium and is widely used to degrade textile industry waste, as it is capable of breaking down all of the complex dyes. Then we have Rhizopus aranus that is capable of degrading uranium, cadmium, and lead, and it is widely used in soils that are often contaminated with radioactive elements. Moving on to yeast. Yeasts are increasingly utilized in bioremediation, particularly for wastewater treatment, due to their adaptability to harsh conditions like lower pH, high salinity, and tolerance to contaminants like heavy metals and antibiotics. And the examples include Yarovia lipolytica, Saccharomyces cerevisiae, Vicia pastoris, and Candida species. So yeasts carry out these bioremediation processes either by accumulating all of the contaminants into their system, that is by bioaccumulation, or by adhering these toxins onto their surface, that is biosorption. Though there are many advantages of using yeast for bioremediation, such as cost effectiveness, sustainability, and everything, there are so many other limitations, such as they may not efficiently degrade complex compounds if their integrity is changed. Say, for example, I just told you that the Candida species that have been used in bioremediation, those are hydrocarbons. In case if you chlorinate those hydrocarbons, then the Candida species, they are ineffective against degrading or breaking them down into smaller elements. They are also extremely susceptible to all of the environmental conditions. even a slight change in the temperature, pH, is going to drastically affect the rate at which the bioremediation is going to occur. And these accumulated metals in the yeast cells, they also pose a secondary pollution risk if not managed properly. And when compared to other chemical treatments, bioremediation carried out by yeast is very slow. Next up, we have bioremediation using microbial fuel cells. Microbial fuel cells operate based on the metabolism of specific bacteria that are capable of transferring electrons to an electrode during the decomposition of an organic material. These bacteria are capable of oxidizing these organic compounds and producing electrons and protons, which is in turn going to generate electricity. In a typical MFC setup, we have three components, which is the anode, cathode, and the electron flow from the anode to the cathode through an external circuit, which will generate electricity. MFCs are termed as a sustainable dual-purpose technology because they not only reduce these organic pollutants, but they also generate electricity. Major microorganisms that are used include Pseudomonas aeruginosa, desulfuromonas acetooxidants, Clostridium butyricum. Now, the limitations of MFC-based bioremediation is that they're very expensive and complex to design, and they are also extremely susceptible to all of the environmental variations such as temperature, pH, and everything. And biofouling issues can also be seen, wherein the accumulation of bacterial biofilms on the electrode is going to affect the rate at which the protons and electrons move across the exchange membrane. Now, the energy produced by MFCs is currently low, which limits their practical applications for large-scale power generation. Next up, we have the use of multi-omics in bioremediation, which include genomics, proteomics, metabolomics, transcriptomics, and single-cell sequencing. Genomics and metagenomics have been instrumental in identifying novel microbes and biodegradation genes. For instance, genomic analysis of the microbial community in oil spill sites identified Alcaniborax and Marinobacter species as primary hydrocarbon degraders. Proteomics focuses on the complete set of proteins expressed by a microbial community in a given environment. And it offers insights into the protein dynamics in response to pollutants. This is mainly done using equipments like LCMS and Magitov. And metabolomics investigates the small molecule metabolites produced by microbial communities. And this branch provides an insight of the cellular metabolic rate, showing how microbes alter their metabolism with regard to whatever pollutant or contaminant is there in their environment. Lastly, we have single-cell sequencing, where it helps isolate individual microbial cells to study specific genes or pathways and connect them to ecological roles within the community. This helps clarify individual contributions within complex microbial consortia and identify the specific strain of a particular compound degrading ability that dominates bioremediation in that specific contamination site or area. With omics, bioremediation can be tailored to the specific microbial community and contaminants at a site, increasing the likelihood of successful degradation. Tools like SIP, stable isotope probing, can trace specific contaminants within a microbial community, pinpointing active degrader species and pathways. Omics data also allows for real-time monitoring and adjustments to optimize conditions for faster degradation rates, such as the precise addition of nutrients or oxygen based on transcriptomic feedbacks. And omics are also adaptable to diverse environments, from contaminated soils to deep sea oil spills. Limitations include they're too expensive, so you can't really apply it to large scale products, and they are resource intensive, and the field applications are limited, meaning whatever is working in the program or the language may not necessarily work in real life. This is a little insight into the recent trends in bioremediation that I got from 67 open access journals. And these are my references, and I'll also be linking them down in the description. And kudos to our professor, Dr. Kavya Bakka ma'am, for being the driving force that she always has been. And thank you for watching.